right, I'm Julia O. I'm an associate professor at the Jackson Laboratory for Genomic Medicine in Farmington, Connecticut. And my lab is a hybrid computational and experimental group looking at different omics approaches and ultra high resolution microbiome analyses to better understand how the microbiome changes in different disease states. So just to give a brief summary of our findings in our latest publication in Cell Host Microbe, which was titled Multiomics of Gut Microbiome Host Interactions in Short and Long-Term Myalgic Encephalomyelitis Chronic Fatigue Syndrome Patients. So our findings really, I think, were the result of the very innovative clinical cohort designed by our collaborators Cindy Bateman and Suzanne Vernon at the Bateman Barnes Center. And so what they did, rather than just recruiting a very broad MECFS patient cohort, what they did was to recruit a cohort of individuals who have been suffering from the disease in short term, so under 4 years, and then a cohort of individuals who had been suffering from the disease for more than 10 years. So a short versus a long-term cohort versus approximately matched healthy controls. And this ended up being a critical differentiator for all of the findings in our study pretty much. And so what we did with these patients is so they collected the detailed clinical data, blood data. They collected very detailed survey data. The patients collected very detailed survey data on their diet, their medications used, sleep habits even. And then what we received were stool samples. So this reflects the gut microbiome. And then we also received samples to look at the metabolome or the chemicals that are present in the blood. So the plasma metabolome. And so these were our major data sets from these patient cohorts. So clinical data, survey, lifestyle data, microbiome data, and then the plasma metabolome data. And so one of the first things we did, of course, was to examine, what is different between the controls versus the short-term group versus the long-term group. And we're really surprised to see that the short-term group had the greatest microbial dysbiosis or changes in the microbiome. And this was true of the diversity, the overall number of microbes that were present, as well as the composition of the different microbes that were there. And this was surprising because in the long-term cohort, their microbiome actually returned to a state closer to that of the healthy controls, showing, I would say, a more normal microbial diversity with reacquisition of some of these low abundance species. And this was surprising because the long-term group, conversely, had the most severe clinical phenotypes, as well as the most differences in the plasma metabolome, despite this normalization of the gut microbiome. And so this gives us an exciting hypothesis that the short-term changes in the gut microbiome, these may be accumulating to have these long-term effects. So there could be damage caused by an initial trigger and then resulting in cascading of events. And so our hypothesis is really here that MECFS progression may begin with the loss of beneficial microbes, which I'll get into in a second, and then resulting in a more pervasive gastrointestinal phenotypes that's later reflected in differences in the plasma metabolome. You can think of, and if I didn't mention this earlier, you can think of these chemicals in the blood as the mediators between the microbes and other cell types in the body, such as the immune cell types, which is studied heavily by our center director, Dariet Odenmatz. So another major finding of our study was that the microbes that were specifically depleted in the short-term patients were these microbes that produce different metabolites called short-chain fatty acids, among which are butyrate. Some other depletions were in tryptophan metabolites and other microbial metabolites that have been linked to mucosal immune regulation. And again, highlighting Dr. Odenmatz's results, these showed they observed a striking immune dysbiosis in different blood immune markers. And so the next step here is to link kind of the differences in the microbiome to the differences in the metabolome, and then differences in kind of the immune cell function. So I would say that the other exciting thing about these short chain fatty acids and these other amino acid metabolites is that they've also been linked to the pathogenesis of other autoimmune or chronic inflammatory diseases. And so again, emphasizing this potential hypothesis is that the microbiome is priming or sustaining an aberrant immune response following the disease onset. So the next major finding of the other paper was not so much a finding so much as the development of a machine learning model to very accurately differentiate or classify the disease cohorts. So you can think about disease classification as identification of biomarkers. So microbes, microbial genes, or metabolites that can very accurately allow you to differentiate a patient versus a control. And so we used developed different machine learning classifiers. And the surprising thing and the interesting thing is that we can take all of the different data sets individually. So the clinical data set, the patient metadata and their survey data, take the microbiome species abundance or their the gene functions or the plasma metabolites, and they create reasonably accurate classifiers. But when you combine all of these omics data sets together, you get a very accurate classifier. And this is really a framework that we think that can be leveraged to study to further differentiate MECFS, severity, perhaps, differentiate MECFS from other diseases with overlapping phenotypes, such as long COVID or neuromyalgia and others. And so, so we also developed this framework for this integrative omics analysis.